Hello learners, in our today's class we have to talk about the antigenic determinants on the antibodies. So what is this concept? For this concept you have to know that if from one person we take the antibody and we inject it into the another person or another individual, it can be of the same species, it can be of the different species. This antibody in the another individual of either same species or the different species will act as antigen. So always remember, if I inject the antibody of one person into the another person, it will act as the antigen. So on the basis of this, when the antibody has to act like the antigen, it has to have certain properties, and those properties are what we call as the antigenic determinants on the antibodies. Means what should be the properties of that particular antibody which has now become antigen, so that now here when it is in another individual it can be recognized by the antibodies of that particular antigen. So in other words, I can say that when this antibody of one person is injected into another person, it will now no longer function as antibody. It will now start to function as if it were an antigen. And as soon as it starts to function like an antigen, the other antibodies of that person will now attack this antigen because it is foreign. So when it will attack the antigen will then automatically be processed, killed, and all those immunological responses will be activated. So what we need to study in this video is that what should be the properties of this antigen so that it can be recognized by the person's antibodies. Means what should be the properties of this antibody so that when it goes into another person it can be recognized by that person's antibodies. So on the basis of that we have classified this determinants into three types, that is isotype, allotype and idiotype. So we have to study the three of them in detail. First of all, we should try to understand that what is the meaning of isotype. Isotype means that condition or the variation in the antibodies in the constant region of that antibody. Means if this is our antibody, so in the constant region of this antibody here, we if there is a variation, then it creates different forms of isotypes. So because of the variation in the constant region and that also the constant region of heavy chain, we have the different kinds of isotypes. For example, we have the isotype IgG, then IgA, similarly M, E, D. These are the different kinds of isotypes of that particular antibody. Now for your kind information, I should tell you that these isotypes remain constant in one species. Means if I am taking two individuals of same species and if I take IgG in them, then these two will be isotypic to each other, which means that both of these antibodies, their CH region will almost be constant. So if I am trying to categorize the antibodies only on this basis, CH basis, then because these antibodies will be isotype of each other, they would not be attacked into the organisms which belong to the same species because they are identical in the terms of the constant region of heavy chain. So isotype is one of the criteria through which we can say that the two of them are identical if the two of them have the similar kind of constant heavy region. Please remember, IgG and IgA are not the isotypes of each other, they are different isotypes. IgA and IgA will be the isotype, but IgG and IgA cannot be the isotype. So this is one individual isotype, this is another isotype, this is another isotype, like that way. Let us try to relate it with an analogy. Suppose I have taken the glass of milk Now this glass of milk, I have suppose taken 3 glasses of milk. Or let's say that I have added milk here, I have added juice here, and I have added suppose lemonade here. So this milk is a kind of isotype. This juice is a kind of different kind of isotype, and this lemonade is another different kind of isotype. This milk and this juice are not the isotypes of each other. Similarly, this juice and this lemonade cannot be the isotype of each other. Similarly, IgG and IgM cannot be the isotypes of each other. But if I'm taking another kitchen where again I have one glass of milk, this milk and this milk are definitely the isotypes of each other. So that is what I'm trying to say. Now let us move on to the another concept, that is allotype. What is the meaning of allotype? So let me rub all of this. There is another common misconception which the people have. The misconception is as follows. We take IgG antibody. The IgG antibody has further 4 types: IgG1, IgG2, IgG3 and IgG4. There are 4 forms of these antibodies. The common misconception is that people mistake the two of them to be isotypes of each other. Are the two isotypes of each other? No, they are not the isotypes of each other. You can consider it another is with the similar kind of analogy, that is, suppose there are 4 glasses. Okay, now one of them contains milk, one of them contains buttermilk, one of them contains curd, and one of them contains suppose cheese. So all of the 4 are the products of milk, but they are not similar to each other. One is not equivalent to another. Similarly, IgG1 will not be the isotype of IgG2. Please remember this. Moving on to the another categorization, that is allotypes. Now allotypes is another important part which we have to learn. Allotype simply says that in the two isotypes, suppose I take IgG from this person and I take IgG from this person. There are two different IgGs, but there will be certain alleles of the genes which are present on the IgG. Suppose here we have allele 1 and here I have allele 2. Both of them are still IgG, but the two of them now, both of them now are different. Although they are the isotypes of each other, but they are not the allotypes of each other. It means that, let's another understand it with one analogy. There is a glass of milk and there is another glass of milk. What we were saying is both of them are the isotypes of each other. But this milk suppose is from a company named MD and this milk is from a company named A. So MD company or MD farm is making this milk or is producing this milk, and the A farm is producing this milk. So in that condition still both of them are milk. That means they are isotype of each other, but they are not allotype of each other because they are not synthesized or made from a similar dairy. So that is the very important concept here which comes for allotype. Always remember that the allotypes are also caused due to the variations in the constant region. Suppose I take IgG example. Due to one allele there is a marker 1, and suppose the similar IgG but the marker is suppose here 23. So these two are the isotypes of each other, but the two of them are not the allotypes of each other. This is very important and which you should know. Also please remember, when I talk about the constant region, the constant regions can be of heavy chain also and can be of light chain also. This is the concept of isotype only. So suppose if I was making this here, wait, let me change the color. If here was the variation, then it was creating the different isotypes like IgA, G, M, E, and D. If here this on also not this, only the constant region of the light chain means this part, these parts, if here are the variations, they make different kinds of antibodies like IgG kappa and IgG lambda. So this is one isotype, this is another isotype because of the variations in Cl region, and these are the isotypes, different isotypes because of the variation in CH region. Then further they can have the different allotypes. Now let us come to the last important one, that is idiotype. What do you mean by idiotype? So let's discuss idiotype here only. Third is the idiotypes of each other. Now idiotypes is another important part because this now depends upon the functionality of that antibody. So mainly the idiotypic portion will be determined by this part, that is the variable part. We have learnt about in our theory about the clonal selection. Clonal selection simply says that in the group of different B cells, a particular antigen will come, and this B cell, if this is exposed to that particular antigen, will make the kind of IgG, and this will also be making the kind of IgG. But suppose this is for tetanus and this is for another important, or let's say another important type of antigen which is coming here. Let's say this is for antigen T and this is for antigen X. So both of them are still IgG, but the functionality, one of the functionality is for anti-X and one of them is acting as anti-T. So these two may be isotypes, may be allotypes, but cannot be idiotypes, can never be. So you can ask again understand it with the help of analogy, that is, take 2 glasses of milk, or let's take 3 glasses of milk. Okay, all the three can be milk, all the three can be again from the same farm or same dairy, let's say a company is producing them. But suppose if they are exposed to different flavors, if we are adding it from our side at home, suppose we are adding, or in this we are adding, let's say honey, here we are adding chocolate, and here we are adding strawberry. So in that condition, although they may be isotypes of each other, they may be allotypes of each other, but they are not at all the idiotypes of each other. So with the help of diagram also we can understand here, as I was telling here. See, so the isotypic determinants will be in the way that there will be the changes in this constant region of either the heavy chain or the light chain. Then if I talk about the allotypic determinants will be basically because of the alleles, of the different alleles of the same gene. That's why we are talking about allotypes. And so they will create the different kinds of allotypes in the different strains. Then there will be the idiotypic determinants, which will be mainly determined by this variable region of light and heavy chain, and because of which the specific idiotopes will determine that specific idiotypes. Idiotopes are nothing but the antigen binding sites where they will come and bind. So this is one of the very important concept which the students often get confused in, and with the help of analogy I have tried to simplify it.